Hello, welcome back. Here's the last mini lecture of the lymphatic system, going over the antibody-mediated immunity. So before I get into the actual process of B cell activation, just a little overview of what we've covered so far. So we start here in this picture, we have the thing that's causing the problem, right? All of the antigens. We've talked about our specific defenses with the cell-mediated immunity. So that was our antigen-presenting cells, activates our T cells, or the infected cells are activated, activating the CD8 cells. And now what we're going to talk about is the antibody-mediated immunity. So we've only looked at half of our two-prong approach. We're going to hit them here, and then we're going to hit them here, and hopefully that's going to destroy the pathogen. So we've covered our cell-mediated immunity. Now we're going to do antibody-mediated immunity. And those are going to be dominated by B cells. So going back to the last lecture, we covered the MHC class 1s and class 2s. So B cells, because they are lymphocytes, fall into the capability of housing the MHC class 2 proteins. So we can see in this step 1 that they have the MHC class 2s, those little blue receptors there on the surface, or these little proteins. And then they also have those antibodies, those little purple Y-shaped things. This is a class of antibody called IgD, stands for immunoglobulin class D. And so they are specific for a particular antigen. I usually like to use the example chickenpox because almost everybody's familiar with that disease. So let's say this is a B cell that is recognizing, it's been programmed to recognize the chickenpox virus. And so that recognition happens on that antibody, the little Y-shaped molecule. So once you get the antibodies bound to your IgDs, those surface antibodies, what they'll do is they process that. And so by processing it, they kind of flip that antibody into the cell, allow for that antigenic piece to get processed into the MHC class 2, and then flip that back up onto the surface. So this is what we call a sensitized B cell because we have the antigen in one of those MHC class 2s, okay? So here's our MHC class 2, so that was called sensitization. When the B cell is presenting the antigen, it's recognized it on the antibody, flop that in, and is now presenting it on the MHC class 2, that's sensitization. The activation piece now is that the MHC class 2 with the antigen kind of hanging in it, a helper T is going to come along. So remember, the helper T got activated by maybe an antigen presenting cell, or it could be activated by this sensitized B cell. However it's going to happen, you're going to get recognition. But it's got to be the T cell, the helper T, that has been programmed to recognize that antigen, because this is the receptor that has to recognize that antigen. It's programmed to be specific, right? That's the specificity of this immune system. So once the correct helper T finds this B cell that's presenting the antigen in the MHC class 2, that will activate the T cell, or it will recognize the T cell. The T cell will then release these cytokines, because that's the job of an activated helper T, releasing all those cytokines, which will communicate to the B cell, and it will trigger the B cells to become activated. Some of them will do those memory cells, which my face is kind of hiding some of those. But a little population of those activated B cells will become the memory cells, lay dormant until the next time this pathogen shows up. But then the rest of these guys are going to go into this pathway here, becoming plasma cells. Now, if you look at that plasma cell, it looks a lot different than a regular lymphocyte, right? So it's got a whole bunch of rough endoplasmic reticulum. And if you remember from general bio, rough ER has a whole bunch of ribosomes stuck to it. And if you remember, ribosomes make proteins. And if you remember, antibodies are proteins. So when you have a plasma cell that's jam-packed full of rough endoplasmic reticulum, its structure matches its function. So you're going to have you basically produce this huge antibody making factory. And I'm not sure, do I have yes, so I wrote the statistic down. When these plasma cells are active and they're actively making these antibodies, they can make 100 million antibodies per hour, which is thousands per second, right? So gosh, they are just pumping these guys out, because what these antibodies are going to do, which we'll talk about in a little bit, is they can go out and they can destroy things within the fluids. The T cells, the cytotoxic T cells kill infected cells, but the antibodies, they can find the bacteria and the viruses that are outside of the cells and neutralize them, so we can help kind of quell this infection. All right, so then this is the step 3, which is division and differentiation. Is once the activated B cells kind of convert to plasma cells, they start pumping out all these antibodies and they just get dumped into the fluids. That's why it's called humoral immunity, because humors are your fluids, or antibody mediated, because the antibodies just go all over the place in your body. So that's how we sensitize and activate B cells via the helper Ts that recognize and release these cytokines and convert those activated B cells into plasma cells, which are antibody factories. All right, so you have this diagram or similar diagram in your lab manual. Hopefully you're reading that as well. So an antibody is a large protein. It's a quaternary protein because it's going to have 4 individual protein amino acid chains. We have 2 heavy chains and 2 light chains. So heavy chains are the purple in this diagram. The light chains are the blue. So we kind of have this heavy chain going in the inner Y and then these short little light chains kind of on the outer edges. So those collectively together make up one antibody. And then within this antibody, there's some things that you need to be familiar with. So these little yellow dumbbell looking things are called disulfide bonds. They are what hold all of these chains together. So they hold the heavy chains together and they hold the light chains to the heavy chains. We can also see that we've got some binding sites. So macrophages can bind onto the tails, if you will, of these Y-shaped antibodies. And then if you remember, we talked about complement, right? So once you have something that's bound, an antibody bound to, say, a bacteria, that complement binding site is going to activate that C1 protein, which then triggers that transduction pathway to produce the membrane attack complex, which will destroy the bacteria. All right. So in this diagram over here, the big orange thing is the antigen, right? So all of this is the antigen, and all the little yellow bits are what's called the antigenic determinant sites. It is the specific molecular shape of those antigenic determinant sites that the antigen binding site binds to. So back when we were talking about blood, I usually drew an antibody like maybe with a triangle shape like this. Maybe this was my anti-A because the A antigen had the triangle shape or whatever. So that's that specific recognition. It is a molecular lock and key, almost like an enzyme in its substrate. The antigenic determinant site is recognized molecularly by the shape of the antigen binding site. And so once those bind, you now have your thing, whether it's a virus or a bacteria, completely covered by these antibodies, which will then allow the immune system to do its job, okay? All right, and there's different types of antibodies. So that general Y shape is the typical of most antibodies, but we'll also see we have some polymers of these antibodies where they combine to be bigger. So you have these 3 classes of antibodies, and I remember them because education-wise, you have to get your GED before you can get your MA, right? GED before master's degree, you got to do that. So that might help you remember. So IgGs, immunoglobulin is what the IG stands for. So immunoglobulin G, IgG, is the largest group of antibodies. These individual single Y-shaped molecules, when your B cells start producing, they don't produce them first, but they're going to produce them at the highest quantity. And these are also the ones that are involved in that hemolytic disease of the newborn when we were talking about that back in chapter 19. When the mom's anti-Rh antibodies were crossing the placenta, these are the small IgGs that could cross the placenta and start attacking the baby's red blood cells. IgEs are bound to basophils and mast cells. So these are what's responsible for a lot of times allergic reactions. So if your basophils and mast cells have IgEs that recognize ragweed pollen or some tree pollen or cat dander or whatever, that's going to activate those mast cells and basophils to release histamine and heparin and trigger inflammation and allergic reactions. So that's the role of IgEs. IgDs are the ones that are found on the surface of the B cell. So when we just talked about sensitization and that little Y-shaped antibody on the surface of the B cell, those are the IgDs. They're bound to B cells and are responsible for that sensitization and activation. All right, so those are all of our single antibody classes. The IgMs, these are the first ones to get released, and they are a 5-pronged starburst. So there's actually 5 individual Y-shaped molecules all hooked together into this one molecule, and these are what we call IgMs. They're the first ones to get secreted, and then IgEs kind of ramp up a little bit later. And these are also our anti-A and anti-B antibodies, these big ones. So if you remember agglutination, so if you had a red blood cell hooked here and a red blood cell hooked here and a red blood cell hooked here, you can see how that would agglutinate the red blood cells and make them clump together. That might make a little bit more sense now when we're seeing these IgMs as these big 5-pronged starburst. And also why you can have a baby with a different blood type is because these large antibodies cannot cross the placenta, but these smaller ones can, right? So when we talked about the hemolytic disease of the newborn, the single IgGs can cross the placenta, but the large anti-A and anti-Bs cannot. And then lastly, we have the IgAs. These have this little secretory piece that allows them to be soluble. And so a lot of times you see IgAs associated with glandular secretions like mucus, tears, saliva, semen, breast milk. This allows for it to be traveling in those fluids. Okay, so those are your different types of antibodies. We're primarily going to be talking about IgGs and IgMs when we talk about this immune response. All right, so that's these graphs here. So let's say you are exposed to something for the first time, and it's going to take a while for your B cells to kick into gear. And if you take a look, it's about 1 to 2 weeks before you get any peak of your IgMs and your IgGs. So let's say it's the first exposure. We're dealing with the coronavirus right now. Right now, nobody's immune to it. So everybody is going to be first exposure. So that first exposure is going to be if you're infected, hopefully you're not. But the IgMs are going to start first, right? So you can see that's the blue line. So they show up first. And then a little bit later, your plasma cells kind of switch gears and they start pumping out the IgGs. And you'll see the IgGs are a little bit higher. So you get IgMs first, but IgGs go higher. And then hopefully after a while, you will recover. Your body will take over and destroy the coronavirus. The cells that were infected will be destroyed, and then you'll get to start in the repair process of healing. Now, if our immune system, and there's no mutations in the coronavirus itself, if our immune system has those memory B cells that it produced in that activation stage, let's say next year this whole virus is going to stick around. We have no idea what's going to happen, but it could potentially come back around. So anybody who was infected this first time around has those memory B cells. So if they get exposed to coronavirus the next year, those memory B cells will respond differently. And you take a look, who gets out in the open first? IgGs, shoo, they just run straight up really high and really fast. So in the secondary response, those plasma cells are pumping out IgGs like crazy. IgMs come along a little bit later and a little bit lower. So that's how you can tell. So if you are not sure if you've been exposed to something before and you say, oh, somebody coughed on me and they were found to be positive for something, you can go and get a blood test. It's called an antibody titer, and they can measure your antibodies. If they find that your IgMs are higher than your IgGs, which is only taking place like right here in this window, then you're likely going to be getting sick because that's the first time that you've been exposed when the IgMs are higher than the IgGs. If it is your first exposure and your IgGs are higher, I'm going to switch colors so we're not doing with all this red. If your IgG is higher than your IgM in your first exposure, it likely means you're on the way down. You've already fought off the disease. You've been recovered. Okay. If you go in and your IgGs are way high, like off the charts high, like what we have over here, it's likely your second exposure. You're probably not going to get sick, and your immune system's already taken over. So that's the main difference, and that's a good thing. That's why we want those memory B cells to be made, because they start right away. Look, almost like time zero. We're getting those IgGs just going off the charts, where here it took time before we start getting those IgGs, maybe a week even, for those B cells to get activated to start making enough antibodies to actually do the work that they need to do to be able to tackle this antibodies or tackle the pathogen. All right, and I think that's it for the humoral mediated immunity. All right, so the rest of the textbook and the lecture material you can work through on your own. If you have any questions, please send them my way. I'll be setting up office hours sometime. At the recording of this lecture, I don't quite know how that's going to look, but hopefully by the beginning of term, I will have that figured out. All right, thanks so much, and I'll see you next time.